Bismillahirrahmanirrahim. The title of our lecture is Autoimmune hemolytic anemia and tolerance, definition, types of induction and mechanisms, and the autoimmunity, which is include induction, mechanisms, diseases, treatment. First, the autoimmune hemolytic anemia, which is type 2 hypersensitivity reaction caused by the autoantibodies against the RBCs membrane proteins, which is lead to lysis of the RBC. And here the RBC, and this is the antigen, which is present on the cell surface of the RBC, and antibody will form autoantibodies against these autoantigens and lead to destruction of the RBC. And the mechanism of the destruction by the type 2 hypersensitivity, which is antigen antibody immune complex, which is lead to activation of the complement and lead to lysis by the classical pathway of the complement, which is the marked membrane attack complex. And the classification of the autoimmune hemolytic anemia, the warm type, which is either primary and secondary, and the cold type, which is include the primary chronic cold agglutinin disease and the secondary cold agglutinin syndrome, and the paroxysmal cold hemoglobinuria, and the mixed cold and warm antibody type. And the third one and the fourth one, the drug used like methyl dopant, which is a drug used in the treatment of the hypertension. The classification of the antibodies is based on the activity at different temperature and their etiology, as we saw, hot type and the cold type, warm type, sorry, and the cold type. The antibodies with high activity at physiological temperature of approximately 37 centigrade are termed the warm type, and the cold autoantibodies act best at temperature 0 to 4 centigrade. Patients with cold type autoimmune hemolytic anemia therefore have higher disease activity when the body temperatures fall into a hypothermic state, as in cold weather. Usually, the antibody becomes active when it leaves the limbs at which point it opsonizes the RBCs. And when these RBCs return to central region, they are damaged by the complement. Patients may present with one or both types of the autoantibodies. Both are present. The disease term mixed type. The causes include the cold and the warm. The autoimmunity is can be defined as failure of the immunological tolerance. So the definition of the autoimmunity is failure of immunological tolerance. What's tolerance? Tolerance is absence of the immune response in an immune competent person. And this is of two types: natural, which is autotolerance, central and peripheral, and induced, which is called also immune tolerance. The central or the natural, which is divided into central, which is currently thymus, that this can be achieved by the clonal deletion, and peripheral, which is achieved by the T regulatory cells, which is called clonal regulation. And the induced, which is induced by the individual or by a person, which is called immune tolerance, which is occurred in the perinatal period, when the antigen is introduced at perinatal period, and this can be seen by the identical twin. And the adult, which is of two types according to those of the antigen, and here the antigen is called colorogenes, and they occur in animals, and it's called low zone tolerance by injection of multiple small doses of colorogene, and high zone tolerance by injection of multiple very high doses of colorogene. And the mechanism of this is saturation of all receptors. Factors importance in tolerance: the time of induction, nature, dose, persistence, intravenous or oral introduction, absence of the adjuvant, cellular involvement. As we see, T helper cell get tolerance faster than B cells, and T helper cell lost tolerance longer than B cell. Mechanism of tolerance: central tolerance by clonal deletion, which occurs in the thymus, and peripheral tolerance by that the lymphocyte inactivation or is called energy, which is different from the allergy. And here the energy, which is in the lymphocyte inactivation by regulation of the lymphocytes that can recognize self-antigen and peripheral tissue through the T reg cell or T regulatory cell. And the antigen sequestration, or called immunological silence, and here the best example is the eye, cornea, and lens. Immune tolerance in pregnancy. As we know, the fetus, which is considered as the foreign antigen, because half of the antigen is paternal antigen, and also the placenta, which is considered as foreign antigen to the mother. So they may be viewed as unusually successful allograft since they genetically differ from the mother. And in many cases of spontaneous abortion may be described in the same way as maternal transplant rejection. And autoimmunity is the failure of the immunological tolerance. Immune cell will react against antigen causing tissue injury and disease. And the autoimmunity occurs normally as a biological process to regulate immune response. Example, idiotype, anti-idiotype network. And this is the natural process that occurs in the body of the autoimmunity. Causes of the autoimmunity is genetic, environmental, and the immune regulation. This lead, when there is a defect in this, lead to autoimmune disease. Autoimmune disease either organ specific, which is mean that the antibody and the lesion occur at the specific organ. Non-organ specific, that the lesion in one organ and the immunological response is described on the another organ. Mechanism of the autoimmune disease: type 2, type 3, type 4 hypersensitivity reaction. Factors that induce autoimmune disease: release of sequestered antigen, for example, I. T helper cell CD4 T cells and its cytokines are more linked to autoimmunity than the T cytotoxic. Modification of the autoantigen by microbes or drugs like the methyl dopa and hemolytic anemia, and the cross-reaction between self-antigen and the microbes like the beta-hemolytic streptococci and rheumatic fever, which is due to similarity between the heart valve and the structure of the streptococci. This leads to cross-reaction between this bacteria and the heart valve. Polyclonal activation, as we see in the Epstein-Barr virus, has led to polyclonal activation of the B cell. Genetic factor, which is association of HLA-B27 with ankylosing spondylitis. Inappropriate expression of class 2 molecules like thyroiditis, which is an increase in the expression of class 2 molecules, which is increase the immune response. Another factor, hormonal factor, as we know that females are more prone to autoimmune disease due to sex hormone, estrogen hormone. Environmental factor, which is molecular mimicry between cell antigen and environmental particles. The last one, thymic defect in the old age individuals, which is thymic will be involved and decrease in the immune system and increase in the autoimmune disease. Here are some examples of the autoimmune disease caused by goodpasture syndrome, which is type 2 hypersensitivity reaction. This is the mechanism for the immune pathogenesis of this disease, which is caused by the autoantibodies against the renal and lung basement membrane, so it's called syndrome because it's involved in kidney and the lung. Goodpasture syndrome is identified as a trial of glomerulonephritis, pulmonary hemorrhage, and anti-glomerular basement membrane antibodies formation. Patients with this disease may present a spectrum of condition ranging from pulmonary hemorrhage, renal involvement, and renal